In this video, we will do a quick tour of the Equine Genetic Calculator software to show you some of the features and do a couple of examples of how to work out the progeny from various horse matings. To start with, you click on File and then Open, and this gives a list of the various groupings that are available. The most common one is the horse colour, which most people will use, but there is also a section for donkey colours. There's a section on the guidelines for heart scores for people with performance horses, and for those who want to know a little bit more about genetics, there's another section on genetic examples. We'll pick horse colour for now. Click on OK. This opens up the mating window for horses. When you first open up this window, it is already preset with a bay sire and a bay dam. To work out what progeny you get from this pair, you click on File, then Calculate, click OK, which produces a report, which we'll just zoom in. And from this particular pair of horses, which are two bays, we get 100% bay progeny. So the DF, or the double factor there next to the word bay, means that it has two genes per bay. So this is essentially a pair of pure breeding bays throwing 100% bay offspring. That was a fairly simple example. We'll now go and do something a little bit more complex. Going back to the mating window again, against the sire you'll notice there is two lines of description. The top line always displays those colours which are visual on the horse. The second line of description shows those colours which are recessive or hidden characteristics in the horse. You'll notice before the second line there's a little slash symbol. This is called the split symbol. So split is a shorthand way of saying that a horse is carrying some recessive characteristics that you cannot see. So likewise with the dam, in this case we have a dam that is visually bay in colour because it's on the top line of the description. And on the second line of description, it is split for no other recessive varieties. So to change the sire and the dam to different colours, we click on the male menu. Now on this list is a list of all the different colour groupings that horses can have. You'll notice there's a fairly extensive list there. And it's actually possible to have all sorts of weird and wonderful combinations of all these different variety groupings. In fact, if you include all the hidden characteristics that the horses may carry, there are actually millions of combinations. With so many possible combinations, it's not practical to have a list of every single combination because the list would be too big to pick from. So Equine Genetic Calculator can handle all of these different combinations by using a different system. What we do is we break the horse's description up into little building blocks, and you use these building blocks from this menu to build up the full description of the horse involved in the pairing. Let's make a new sire for our mating. So we click on male to change the sire. We'll look at the first grouping here, which is for light bays, browns and blacks. So we click on that and it opens a variety combination window. So what this gives is a list of every possible genetic combination involving the colours of light bay, brown and black. So amongst the list here, we'll pick one that says, how about we pick a bay single factor split for brown. So what that means is we have a visually bay horse that carries one gene for bay, and it also has a recessive characteristic that it can potentially breed brown horses as well. So it's visually bay, but has the potential to breed brown horses. So to select that, we go Select and Done, and we look at our new description here. So now on the sire description, in the visual line, it says it is visually bay. On the second line, it says it is split for brown. So if you want to change the dam, you click on female. We'll go to the same section, and this time we'll pick just say, let's say a bay horse that is split for black. Select and Done. So again, down here in the dam, we have a visually bay horse, which is carrying a hidden gene for black. So it's bay split for black. Let's add something else to it. Why don't we pick, say, Sabino? So in this list and several lists like it, you'll notice the very first option list says normal. What normal means is that the horse does not carry any genes for whatever else is on that list. So if you pick normal from this list, it means the horse is not Sabino. So let's pick a single factor Sabino, which means is it as a horse with a pied type pattern, and it only has one gene for this particular colour. So you go select and done. And you'll notice there now the sire says it's a bay Sabino horse. They're both single factors for those colours, and it's split for brown. So to work out what you get from that mating, we go File, Calculate, press OK, and we look at our report. So in this report, there's 8 different colours we can get from that pair of horses in various percentages. In fact, in this case, they're all 12.5%. And looking at the first one, we can have brown single factor split black. So that means we have a visually brown horse carrying one gene for brown, and it's carrying a recessive gene for black that you can't see. The next one is a brown Sabino, also split for black. We get bay that is split for brown, bay Sabinos split for brown, bays split for black, bay Sabinos split for black, purebred bays, and bay double factor Sabinos. So there's 8 different types of progeny from those 2 horses. That's an example of how we can change the colours on horses and work out what progeny they get.